It's my pleasure to welcome and introduce Dr. Jan Schuemann for today's IMO seminar. So Jan is an associate professor as well as the associate director of physics research and the head of the Multiscale Monte Carlo modeling lab at Harvard MGH. He's got his PhD in Japan, then went on to do a postdoc in Japan and Taiwan before moving to MGH in 2010. There he rose through the ranks of instructor, assistant professor, and now associate professor, and is also the current vice president-elect of the Radiation Research Society. So Jan and I actually teamed up for a joint mini symposium at SMB on math modeling for radiation research, and also at the annual meeting at the Radiation Research Society later this year in Hawaii. So looking forward to that for the science only.
Yes, of course.
So Jan is very interested in multidisciplinary research and multi-scale Monte Carlo simulations and understanding cell scale effects, and of course, all with a focus on radiation therapy and radiation oncology. And with that, I'm quite happy to hand it over to Jan and welcome again.
Good. Unmute as well. And thank you very much for that introduction. I actually got my PhD in Taiwan, but I was working in Japan or studying in Japan. The experiment was there, so I went back and forth a lot. So basically, either term works. Well, thank you very much for the kind introduction. I've always been fascinated by how concepts work across multiple scales, how they are connected. And this has been exemplified very nicely in this fascinating movie that I loved in my high school time. I don't know if anyone of you knows that. Anyone actually can see some of you. So if you know this movie or feel like you know it, raise your hands. It's a little bit older. So it's from at least one of them. Wonderful. It's a little bit older. It's about 10 minutes long, and it goes through 40 orders of magnitude, introducing different concepts along the way. And well, we've requested that a lot of times at our physics class, because it was fascinating and I find it really, really amazing. But it does show a little bit what we are working on, trying to connect the different scales. And it's called Powers of 10. In German, it's called 10 hoch. So have a look. This poster is not the best. I did like the images the best here, but they did mess up a little bit the sign here. So anyways, it's a great movie, although a little bit old, but go have a look. I don't really have any important conflicts of interest. But this movie also shows, if you ask a physicist, I think typically we would say we can explain really everything, because everything happens for a reason, and that reason is physics. Because if you go down to the details, you should be able to describe the whole world with physics. There's a little bit of a problem because obviously it's very hard to describe everything, like the onset of biology with physics. There's a bit of a gap here between what happens at the atomic scale and how life evolved. And so it's not always straightforward to describe everything with physics, but that doesn't mean that we don't use physics to apply to biology. For example, shortly after Marie Curie found radiation or discovered radiation, we used it in tumor treatments. And this is a little bit more of an advanced tumor treatment here, but it was very quickly moved towards treating tumors. And of course, it's not just a big gap in between physics and biology. We kind of know what happens in between. At the physics scale, we have direct interactions of the radiation with the tissue or DNA material. Then we have these excited atoms that the initial radiation excited and dissociating and creating different oxidative species, mostly oxidative because we're mostly in water. And then at some point, chemistry takes over, where these molecules that are created then, or reactive species that are created, can interact with each other and form new species or potentially damage the DNA. And so this is all the damage induction side of the equation. And then biology, somewhere in the order of 10 to the nanosecond or maybe millisecond area, biology starts to occur, trying to undo all this damage that physics and chemistry incurred on a cell after radiation. And so it propagates the DNA damages, it potentially results in some kind of repair processes or failures thereof, and then that can be influenced by different aspects like the microenvironment or what kind of cells you have, and result in all sorts of downstream biological effects. Now, we can visualize this very nicely. You can see here is a physical track. The red is electrons, this blue is a proton, the yellow is like the initial ionization excitations of species, and then you can see migration of chemical species around. So this is basically all the stuff we can model for the DNA damage induction, and then biology starts to repair all the DNA, and you can see here a plasmid ring of DNA. And this can be extremely well described using Monte Carlo. I don't know if you all know what exactly Monte Carlo is. So it's I know everyone always says, oh, Monte Carlo is super accurate, so everyone accepts that. Monte Carlo actually just means well, it actually was coined in the Second World War based on research in the weapons project in Los Alamos, and was named after the Russian roulette in Monte Carlo, where there was a lot of random number generator, well, which is a simple random number generator. Of course, you can also just roll dice or have other random events. And therefore, Monte Carlo really just refers to any procedure that involves sampling from random numbers. And that is a very powerful method to describe radiation, because or particle movements through any material, because there is a probability of certain events happening. And if you then look at, for example, a proton passing some other atom, you have a certain probability of the proton is passing by, or having all sorts of different interactions with this nucleus or atom. And then the integral over all the possibilities should, of course, be 1. So something will always happen. And so this method is very powerful, but it's also quite time consuming. So one of the downsides of Monte Carlo is really that we need a lot of statistics to get a good hold of the answers that we're looking at. And so for clinical purposes, Monte Carlo for a long time was way too slow, despite the fact that everyone knew that if we use analytical mechanisms, we are compromising on the accuracy, but the time to wait was just too much for the clinicians to and the treatment planners to get accurate distributions. And so a lot of time was spent using analytical dose calculations. We still use mostly analytical dose calculations in the clinic. But you can see that there are differences. And if you look at the difference profile here, this is a very typical beam that shows why Monte Carlo is useful or how it can describe things better. Because you have in this proton beam coming from the top here, you have a very high density gradient with this bone along the beam line, and then air cavities left and right. And that just results in a lot of statistically more scattering in the bone than in the air. And therefore you have scattering out of the bone, and resulting in this lower dose behind the bone structure and a little higher dose around it, because you scatter into this. And if you are targeting an organ at risk, this can become potentially important. So we've done a lot of studies on that part. So Paganetti actually estimated the importance of Monte Carlo for predicting the range uncertainties. So all these differences we knew that analytical calculations are not as accurate, and so we assigned a certain range uncertainty. In most clinics, I think we use about 3.5% that we just add of healthy tissue that we irradiate to make sure that we hit the actual target that we want. And Harald Paganetti estimated where these come from again after the initial estimate only included 2 factors, but we knew a lot more by now. Well, this is also already 10 years ago. And he estimated that basically, especially for heterogeneous tissue, we can greatly reduce the uncertainty and have a general uncertainty of about 2.4% instead of 3.5%, or in heterogeneous tissues even much higher. And this can become important because you can then reduce your margins and save healthy tissue that you know is potentially healthy, but you include for ensuring tumor coverage. And so it's obvious that Monte Carlo is very powerful, and it's been used in many different applications, actually in your stock market predictions, traffic control, but in radiation therapy also in many different areas. We typically use Monte Carlo in phantom simulations or treatment planning, treatment plan evaluations. And especially my group especially looks at the smaller scales, trying to understand what happens in the cell. Now, when I talk about Monte Carlo, I typically talk about Topaz. Topaz is a Monte Carlo code that we've developed over the past 12 years now, which really the idea behind Topaz is to wrap GN4, a general purpose Monte Carlo system that was developed at CERN for particle transport, and make it useful or more intuitive to use for the field of medical physics. And this idea actually was first hatched by Joseph Hoops, Joseph Pearl, Bruce Fadigan, and Harold Paganetti, and then more and more people started joining the team and worked along this for a while now. And the idea really is to make it easy, to make it a well-tested code that users can use, because a lot of times if you have any experience working with Gen4, you have to write C++ code, you have to compile your own code. Oftentimes, large parts of code have been passed around, and everyone adds their own if clauses and little pieces of debugging code that then gets passed along. And so it gets very messy. But the idea here is that we have a clean code that's well tested, and all you do is add parameter files that define basically anything you want to create, geometry, particles, graphics, and so on, and really try to make it robust. So you can't make line ordering mistakes and so on. And it looks very simple. All you do is every line defines one property. For example, here you have a box that you want to create. It's a geometric object, and it's filled with air, and it's about 4 meter half length. Anyways, it's a Gen4 term. So it's very easy to define properties line by line, and it has a lot of error catching that will be, once you run it, it will tell you that you're probably doing something wrong and tell you exactly where you're doing it wrong. So that's the whole idea. And with this concept, you can do a lot of great things. This is, for example, our double scattering module. You can see here the rotating mod wheel. The beam comes in, is modulated. It goes through the scanning magnets here. You have a second scatterer, ion chamber, snout, collimator, aperture, and compensator. And at the end, you saw the water phantom there. And these are all built on the fly. Even the movement of the video is all controlled by these parameter files. So it's a really powerful system, and we have had a lot of success, or a lot of people have really liked working with it. And one of the things we've done, as I said, Monte Carlo really is just anything that rolls a random number, which also means you can get basically any result that you want. But we do want to make sure that when you simulate something, you want to get actually the real world experience. So you want to actually simulate what you're delivering. And for that, we've done some comparisons to our daily or weekly QA measurements. We've done comparisons to many other experiments that we've done. And we find that overall, we're very much describing our experimental data. You can see here the difference between proton and Topaz simulations and measurements. And so we were very confident that we describe what we simulate, or our simulations describe what we actually deliver. And so we applied our Monte Carlo simulations to initially treatment planning systems. Here, a study looking at the uncertainties that trying to understand how much, assuming that Monte Carlo has an uncertainty of 3.5, and we compare it to the 2.4 as hard estimated, and compared to the 3.5%, how is that enough? How well do we do here? And you can see again a field of proton field with Monte Carlo and analytical dose calculation. And the difference, the interesting thing here is that if you sum up the differences, it's actually nearly zero, because all that we do is we redistribute the protons, but you can see that there are large fields of uncertainties of different under or overdose, basically. And so if you do the root mean square of this, it's nearly 5 millimeter difference. And we estimated that for different treatment sites and found that, again, not very surprising, for heterogeneous patients, we see a lot of spread in the actual range uncertainty, whereas for homogeneous patients, we can actually reduce the range margin without any downside, because our analytical dose calculations perform very well. Now, this is for single fields. Typically, in a treatment, you have multiple fields that are delivered, and of course, that mitigates a little bit these range differences that are potentially occurring from the analytical dose calculations. And so we looked at the full treatment plans, and we found that for the dose distributions in the patient, really it's all relatively accurate. We are within 5%. Analytical dose calculations typically underestimate the scattering, so it's typically a little bit lower. And again, you see for heterogeneous patients, we have a higher uncertainty or a higher spread. But you can also see if, for example, for lung, if you look at the tumor control probability, you have you can have up to 10–11% under a lower potential tumor control probability, because you're at the steep end of the dose response curve. Now, the good news is most treatment planning systems nowadays actually include a Monte Carlo option, or at least a validation with Monte Carlo, so that these uncertainties will get a lot smaller. But you see that biology actually does matter. If you include some biological factors, your dose differences may become bigger. Now, if we think about biology, a lot of the biology that is important for radiation therapy is the cell killing. And we want to basically eliminate the tumor, right? So the most prominent model that describes cell killing with radiation is the linear quadratic model that describes the survival fraction for any given dose, depending on the tissue parameters alpha and beta, or the alpha-beta ratio, which basically gives you the sensitivity of the tissue. You can also see that it depends a little bit on the radiation quality. If you have alpha particles, you have a much steeper curve here. And this is basically just a model trying to describe these data. And we have a lot of these data for different cell lines from photon cohorts. There are also several different models that have been proposed. But we're looking now at proton therapy, and there you want to use the vast amount of data that is available from your photon experiences of photon treatments and photon biology experiments to predict what happens for your proton fields. And for that, the so-called relative biological effectiveness concept has been designed, which basically just is the ratio of doses that you need to deliver to cause the same biological effect. But you can see that this is not a constant value. It actually changes, for example, with dose. We can simply conceptualize that if you give about 100 gray to anything, basically nearly everything will die. And so the RBE would be 1, no matter what radiation quality you use. But yeah, so it varies with dose. Typically, it's assumed to vary with dose, the LET, so the radiation quality, and the tissue sensitivity. Right, but in the clinic, we actually use a constant RBE of 1.1. This is not so bad because we really are mostly concerned for the clinic at the center of the SOBP, and if you and there the RBE is about 1.1 because this is where you treat your tumor, but there is a lot of variability, and as I already shown, RBE depends on different things. I've already mentioned the tissue and the radiation quality. I've shown that it depends on the dose, but I would like to give a few more examples. One of my favorite examples is actually, it depends on the modeling approach. And this is one reason why it's very hard to convince clinicians to use your RBE model, because there are many different RBE models. And depending on which one you have, which one you use, you can have quite large differences in the prediction of your biological effectiveness of your radiation for the same dose distribution and LET distribution. So the LET is the linear energy transfer. It's how much energy is deposited within a certain amount of track length. And yes, you can see, however, that they all depend on the LET. They all go kind of along with the LET here. They also depend on the alpha beta ratio. And this is actually an effect that is oftentimes not as well considered in treatment planning, because what we are looking at is we're having a dose of 1.1. And if we assume a tumor radiosensitivity, which is typically not very radiation sensitive, so rather resistant, then the distribution actually looks quite similar. You can see, if you look closely, you have this increase in biologically effective dose or RBE weighted dose at the end, following this increase here. But if you then assume that your tumor is radioresistant, but the healthy tissue around it is actually a typical healthy tissue of an alpha beta of somewhere between 2 and 4 grade for most healthy tissues, then your potential biologically weighted dose is much more prominent in the healthy tissue around the target. Now, you could use this to optimize a treatment plan, but again, you can see that it depends a lot on the model, but all of them follow the same LET trend. So you can see that everything is very related to LET, and we like LET because it's a physics parameter. We can actually calculate it, and it's independent of the model. So Clemens Grasberger did a study where he looked at different LET plans. So you try to make the same dose distribution with 2 different plans, and you can see if we change the way we deliver, we can actually move high LET regions from outside the treatment field to inside the treatment field target region here. And so from a biological point of view, we would consider this field to be likely more beneficial, because you have higher biologically weighted dose inside the target and less outside. Now, this concept can be used to move towards LET optimization included in your treatment planning, which reduces the effects of RBE. We know, for example, we don't really know what our tissue sensitivities are at somewhere between 2 and 10 gray typically. It's uncertain, but all of the models, all of the data shows that basically with increasing LET, no matter what your alpha beta ratio is, your RBE increases. So if you optimize your LET, low for healthy tissue, high for normal tissue, the other way around, high for tumor tissue, then you will get a potentially better, but biologically better treatment plan. And Jan Unkelbach actually did some very nice studies here, where they re-optimized treatment plans to move the high LET region here that would have been delivered to the brainstem outside of the brainstem for a typical meningioma case and a skull-based chordoma. And this is a method that works very well. It always comes with a little bit of a downside, because you have to put the high LET somewhere. So you may have other hotspots, but you can choose where you put those. Now, we thought this should give us very clear clinical data. So let's look if we can find correlations of these high LET regions with biological side effects. And here, Drosela actually looked at nearly 120 patients. We only found 4 symptomatic changes in the brain after radiation, and only one of them, the one shown here, actually correlates with LET, where you can clearly see in the high LET region or the high biologically weighted dose region, you have the potentially necrotic region in this patient. And if you look at this, you calculate your dose, this is your physical dose, and then your biologically weighted dose, you can see that this region in the necrotic region actually is shifted much higher than in the target region. So this could potentially explain why this patient had a necrotic onset there. There's of course some uncertainties in the modeling, and you don't really know what your alpha beta ratio is, but it may be related to this. However, it also shows that it can't be the sole contributing factor, because we only saw that in 1 out of 4 cases. So the LET and RBE variations may actually be a lot smaller than the patient specific variations of radius sensitivity. Similar, a study of 50 proton patients, again 50 patients this time, where we found necrotic regions in the brain after radiation therapy in a 12-year time span. We looked at different, we identified them on CTs and MRIs, and looked at if we can correlate the region of necrosis with LET. And we found that overall, the mean, there's basically no difference. On average, there's no difference in LET in the necrotic regions. But there are some patients where potentially these may have had a specific radiosensitivity or be more radiosensitive to these LET variations. So if we could identify these patients pre-treatment, then we could potentially adjust the patient treatment plan to help reduce the potential side effects. Now, one of the problems with RBE is that it's actually a very generic number that's derived from typically clonogenic survival data or similar cell-based data. It increases with decreasing dose. This is actually mostly the highest dose, of course, is in the tumor. And so in normal tissue and organs at risk, you have typically lower dose. It increases with alpha beta ratio, which again, with decreasing alpha beta ratio, which again, typically is in the organs at risk, and it increases with LET. And because of these beam configurations, like you might have seen, the high LET is often also in organs at risk. So the question is, how relevant is this in vivo cell survival data for RBE? How relevant is it for tumor control? It's probably quite relevant, because tumor control really we do want to have related to cell death. But in normal tissue toxicities, it's definitely also related to cell death. If you kill too many cells of your any organ, then it's not good. But it also can potentially be related to many other things, like mutations or other signaling pathways that have downstream side effects that are not wanted. And so it's really, RBE is really a generic concept which gives you one value for all the stuff that happens in biology. And I said it's determined from some kind of colony formation, foci formation, micronuclei formation. And it really is a bit of a mystery how we get to RBE after biology occurs somehow. Now, to really look at or understand where the differences of radiation response of different tissue, different cells comes from, I think we need to look at the cell scale and really understand what's happening at the cellular scale. And just to illustrate, it can be, we can very easily see, for example, why higher LET would be more detrimental to cells. If you can see, here is a proton track simulated with electrons that come off the proton track. It's basically the interactions of the proton track in a cell or in a tissue. And you can see there are some interactions. This is a low LET track. If you go to lower energy protons or high, or here actually we're using carbon because it's more dramatic, and higher LET, you can see there's more and more electrons created around this particle track. You can see that there is a lot more energy deposited in this around the track. And if you just look at it head on and zoom in a lot, then you can see there's this large halo of electrons that is around each carbon track here. And if you overlay that with a DNA, here just a simple nucleosome, so 200 DNA base pairs, you can see that if this track passes through your nucleus, it will damage a lot of the DNA that is in the path, and therefore cause a lot of complex damages in this DNA. And this is what makes it hard for the cell to repair, whereas these simple tracks that go through will only damage very localized small, do smaller damages. And to really investigate these effects, we developed Topaz and Bio, a code that really wants to look at the effects at the cell scale. And the idea is that the clinical endpoint of interest really is not the dose that we deliver, that we can calculate very nicely with our Topaz Monte Carlo, but is the biological effect. And so we want to study the biological effect, and the fundamental unit of the biological effect is the cell. Now, a cell is a rather complex object. If you ask a biologist, it probably looks something like this. It has a nucleus, it has mitochondria, a Gvalue body, and all sorts of other things that are around there. If you ask a chemist, it's typically more something like this. It's this body of reactions and migrations and reaction centers that happen. And if you ask a physicist, it's basically just this big box of water with some cross-section. So that's how we typically describe the cell. If you ask a modeler, it's probably just some kind of integral of a certain function. Now, if you leave the modeling out for a minute, the idea what we want to do really is to adjust. I don't know if you can actually see, how many of you are physicists and can't see a difference between these. Hopefully you can all see that they are different. But what we want to do is adjust the model of the physics to more represent what actually happens or how a cell actually looks like, and then include chemistry and biology to describe what happens at a cell level. And so for that, we started out by looking at developing a lot of geometric models. We have developed different DNA models. Typically, we have either spheres or cylinder cutouts to represent our DNA molecules with a DNA double strand wrapped around histones here. Then these are arranged in certain forms. Here the cutout, again, wrapped around the histone. We form these chromatin fibers, and then those are arranged in this, this model in a Hilbert filling curve to fill in a fractal walk the entire nucleus with the 6.1, not mega base pairs, but giga base pairs of DNA. Another method of describing the whole DNA is the high C implementation. It's a relatively recent microscopy method that actually can tell you how likely different parts of the DNA are in close proximity. They basically break it all up and reconnect them, and then see how often things reconnect. And they form these topologically associated domains that represent certain sections of the DNA. And with this method, you can actually measure and then recreate different types of cells. And so we're now working on actually filling all these tads with DNA, again with different structures of DNA, to really be able to describe different cells at the DNA scale, and then build our whole cell. So you have different models of the nucleus, then you can fill a whole cell with it. You can add the mitochondria. The mitochondria can again have different substructures because they are the only ones that have extra nuclear DNA. So we included those. You can have different shapes of your cells. You can have basophils or bone cells here put in a matrix with the osteoblasts, osteoclasts, and sites. And we're looking at trying to figure out, for example, here, how these interplay with each other, how these potentially are differently affected by radiation, which is why we sometimes see bone loss after radiation. And you can, of course, assemble them to fill out a whole cell dish to represent what most biologists see in their experiments. We also have had a large interest in looking at neurons. So for that, there is a great database called neuromorpho.org that has over 100,000 different neuron images, and you can just download them and then you can read them in with Topaz. You can get these different neuron structures. You can assemble them into neural networks. You can add astrocytes to have the helper cells around them as well. And then we're trying to understand how radiation potentially impacts the cognitive function or the signaling between neurons in different radiation scenarios. For example, we have one study where we're looking at the sense of smell, how that is affected for some, especially pediatric patients. Then these neurons, of course, have themselves additional substructures. There are dendrites on the neurons. They have different spine, or the dendrites are these arms that come out, and each of these dendrites have spines on them with different maturity. So they have different structures or different types of spines. These again can have different substructures inside them. For example, some polyribosomes. You can have the membrane of the spines simulated is the lipid bilayer with these membrane proteins, because some of the signaling could be impacted by impacting these proteins. Again, the mitochondria can be simulated, and the whole nucleus can be modeled here with a slightly oval shape. So you can build your complexity in these simulations depending on the questions you really want to ask. Right. And so that's all the geometry side. And then the chemistry starts. I already showed you this video, but this shows the initial physicochemical state, so all the initial ionizations that happen, and then the movement of chemical species or radicals. The limiting part of this is that most of it is still, in most Monte Carlo systems, it's modeled as a water environment. Most of your cell is water, but there are certain important differences in a cell. And so we are working on including these differences, including scavengers that happen in a cell, because in water, these radicals can live for microseconds or longer, but in a cell, anything will be scavenged within a few nanoseconds. And a new method that we are using is the so-called independent reaction time. Or actually, it's a very old method, but we are including it new in our Monte Carlo, which basically, instead of tracking all these steps that we saw before, and just uses reaction times and radii between different potential reactive partners, and then just sorts them and basically executes one and resamples this table. This is a very powerful method that we've applied to do different things. One thing we always do is we want to make sure that we're describing the data. So we always try to find data that that we can fit that we can either fit our data to, or that we can validate our data with, our simulations with. One of the problems often at these scales is that the data it's very hard to find very good data for these simulations. But we do, Gvalue is a very, very useful chemical value that is measured, very well measured. And so we simulated that. We were looking at different time structures and how that impacts the development of the presence of different radicals. We looked at long-term reactions here, and the fricadosymmetry, which is another frequently used measurement. And we looked at different structures, different DNA damage simulations, including the chemistry. And one thing that often has not been considered is actually changes in the temperature, which for your simulations, you can see that there's quite a bit of a difference if you increase your temperature. And many times when we simulate things, most studies have actually neglected to include potential temperature effect, because sometimes cells are imaged at or irradiated at room temperature at 37 degrees or even at cat cold. Now with that, I've shown you we're working on a lot of the damage induction side. We've done a lot of can simulate this quite well. What about the biology? What about the repair? It's rather complex. There are many different things that happen. There are different types of repair lesions that can happen and different proteins that are recruited. So we've tried to simulate it again with a complex system where we have here the Damaris code that was developed in Manchester. And they did a great job in, again, looking through the data, through the available literature, to come up with recruitment times of different proteins for different repair pathways, and then see use the damage structure that we have of location of the different DNA damages and the subdiffusive motion of these DNA damages, because of course they're not freely moving around these DNA damages, but they're constrained by the DNA strands that they're connected to. And with that, you can actually simulate the movement of these different breaks that you induce and the repair processes. And then you can see some of these disappearing, and so then they would have been repaired, or some of these may stay misrepaired. And with that, we've used a lot of data to fit the model and then to test the model. And you can use this to really study different things, correlating biological endpoints, like the looking at RBE, for example, correlating dose and LET. You can try to look at how different cell types may be affected, because they may have a knockout, they may have a repair pathway that's missing. And you can see if your model can actually predict what would happen to these cell types. And then we can look at different effects. I've shown you this high C mapping. We're working on actually getting the DNA representation to not only have just DNA representation, but different density, different complexity of the DNA packing, because in the cell you also have, depending on the cell phase, different density of your DNA. It's called so-called hetero and euchromatin regions, and this also can impact your result. Here's a short example of what we applied this to. We used our cylindrical cutout model for the DNA here, and typically you define single strand breaks. It's just a break on a strand, and then double strand breaks are if you have damages within 10 base pairs. These 10 base pairs, actually, chemists explained to me that this is about the distance where, if it's longer, then the DNA attraction to itself can keep the strands together. So that's why at about 10 base pairs, they just basically break apart. Anyway, so if we include these, we simulate different single and double strand breaks, and then apply that to predict the onset or the observed dicentrics that we can see, or micronuclei formation. And we actually can get quite a good fit to the experimental data. So this is quite powerful. I think we can do a lot of studies. A lot of these are actually best if you do relative studies, because a lot of the uncertainties cancel out. But overall, I think our modeling, what we're trying to do is really include in our Monte Carlo modeling the physics, physicochemical stage, the direct and indirect effects. The indirect is also chemical effects. Include the cell repair modeling to really understand what's happening there, and then come to an endpoint eventually to be able to predict how will a patient potentially do. So this is a bit on the outlook. We're working to get towards the endpoint calculation. So if you're interested to learn more about it, for the macroscopic simulations, you can go to Topaz, readthedocs.org. There's a lot of information there, also how you can get it. For Topaz and Bio, it's topazandbio.readthedocs.org. Topaz and Bio is open source. Topaz will be open source later this year. It's free to use right now. We're working on ironing out some ways of how we want to handle all the potential pushes and so on from potential users. And so with that, I'd like to thank all of you for listening, and all the collaborators of especially the Topaz and Bio collaboration, the team at MGH, other people that have helped and given advice, of course, our funding agencies, and of course, all our users that are using Topaz and help each other on our forum. I think with that, I thank you all, and I am ready for any questions you may have. It's fun that you're muted while you're clapping.
All right, let's see. Does this work now without feedback? Yes, man, we are so technically skilled. That's awesome. All right, Jan, thanks so much. That was excellent. Do we have any questions? Well, I've got a ton now. Sunny, go ahead.
I've got a couple, but it's more to do with model calibration. So, bridging all of those scales is, first of all, kind of incredible, but obviously very ambitious. One of the things that I worry about is error propagation. So, mistakes at one scale affect the next scale, affect the next scale, and a small sort of error becomes a huge error. How do you deal with that sort of problem?
Sorry, I couldn't hear you. I can't answer that.
I don't know if you heard.
No, I'm kidding. It's a tricky question. That's why. Yeah, no, it's a very good question. We actually did some studies to show how simple different assumptions, like slightly different assumption when you cause a double-strand break or which chemical model you use can have large effects on your overall number of damage that you predict or outcome that you predict in the order of like 20 to even up to 100% difference. So we're actually hoping to work on a project looking exactly at the different errors, the different uncertainties of all the different parameters. We know our physics parameter differences, uncertainties at the cross sections at very low energy is still in the order of like 10%, I would say easily. There are, the chemistry again has certain uncertainties, so it all does propagate, but in the end, there is, you always, that's why I said relative studies are much better. If you have if you kind of normalize your effect size to an outcome, and you predict, for example, the number of double-strand breaks in one setting, the relative effect, I think, if you change one thing like the different radiation type or so, that can be quite well predicted. The absolute effect is really hard because you have all these uncertainties in actual prediction of numbers. But we are hoping to do a very systematic review of this, and so.
Yeah, I know that was a tough question, but it's a question that we have to sort of wrestle with, right? And I think one of the ways we've tackled that sort of complexity is actually through exploiting homeostasis. So the fact that the non-treated system should be homeostatic, although parts of that tissue could still be infected by the tumor, and so you're trying to balance that somehow. But the non-tumor part of it, a certain behavior should occur there, and what normal is, and in the absence of any transformation, what should occur. So you have this kind of ground truth that you can control a lot of that error with. Do you see what I mean?
Yes, it's a bit tricky when you're dealing with a system that tries to explain the effects of radiation, right? Because we're looking at damage induction, damage repair. So the homeostasis is not really, I mean, we assume that to be stable. But what we use typically is we have, we have relative studies. And so then, yeah, I mean, it's kind of similar, but it's a different normalization point.
Okay. I have a question before I go with Miriam. So looking at, so we're bridging scales between the nano, micro, and macros, all these scales together. I'm wondering if my research questions deal primarily, say, in the macro scale, looking at on a lesion, for a particular lesion or organ system, the whole patient. And okay, you have the micro scale models. Well, the question is, do you have any plans on coarse graining that for computational simplicity, for especially those questions, like if I have a question on, okay, just what is the radiation going to do for the patient? I'll just leave it at that.
Yeah, I mean,
You know what I mean?
I think so. As I said, you have to carefully choose what you want to simulate for the questions you want to answer, because computational time is still a problem, especially if you look at DNA scale simulations. You can't simulate a whole patient treatment with these DNA scale accuracy. That'll just take forever, even with the best computers you can have right now. But we are looking at, for example, informing the next level based on our smaller scale simulations. So we have DNA or cell scale, DNA scale simulations that we use to inform our microdosimetry simulation. So we do some microdosimetry to then use microdosimetry in getting a better model of biological outcome at a larger scale. So you can kind of scale up, and each level kind of informs the next level, and thereby you can have a very reasonably fast and inclusive approach to determine radiation effects based on all the different scales, informed by different scales.
Right.
But for example, one thing I would like to do, or we are working on, is having this model where you can just have a treatment plan that you deliver. You get the macroscopic plan delivered, you get the field, and then you can select in your patient a few regions where you say, okay, I'm interested in what happens here, and then you can potentially adjust the parameters for that. What's the microenvironment? Is it hypoxic? Is it something? So you change how your cells, what cell type you have, for example, tumor or neurons or so, and then select a few of these points, and then you kind of scale down to what happens at these small scales. So that's something we're working on, to really have a macroscopic to microscopic picture that we try to study them, and hopefully can make useful conclusions from that.
Thank you.
Yeah, so Jan, one of the things we are really interested in is trying to simulate the overall population response to radiation, and linear quadratic model, everything is nice. It gives you survival rate at a macroscopic scale, if you wish, or probability on an individual cell level. But one thing we are struggling with is, when do cells actually die? This becomes really important for fractionated radiation, right? If your cells are stuck in DNA damage repair for 2 weeks, we don't need to irradiate every day, right? So one thing we haven't really quite figured out yet is what are the dynamics and the timelines of cell repair attempts as a function of dose. I was really excited to see your DNA double-strand break dynamic simulations and your correlation with gamma H2AX and stuff. So are these approaches calibrated enough for different cell lines that we can get an understanding on the distribution of cell dying times?
I'd say optimistically, yes.
Again, please.
Optimistically, yes. I mean, yes, we do have a propagation of the DNA damages, but basically after 24 hours, we say everything that's not repaired yet is just going to be unrepaired. For now, I mean, we can potentially extend it. Sorry.
Right. So sorry to interrupt. So this might be true for 2 gray, right? But if you give larger doses, you have potentially larger repair times. They can still get repaired. So can your simulations do that as a function of dose? Do we have data to calibrate that?
I don't think. I mean, the function of dose in this aspect in our model would only be how much damage there is, how close they are together, how much it does impact the misrepair or the unrepaired cells at 24 hours. We just basically chose a 24-hour cutoff at the moment. We could potentially extend that. I don't know if we have enough data to really know how that yeah, what happens then? I mean, most models assume that everything is repaired after 6 hours, right? So that's why you have potentially daily fractionation or morning, evening fractionations or so.
Right.
But yeah, for high doses, maybe.
So from your model, you could potentially get a distribution profile of when cells exit the repair phase instead of assuming like it's a 0-1 at 6 hours.
Yes.
That's cool. Definitely have to talk about that.
Are there any other questions, either on Zoom or in the room?
I did have a question. So so far I've seen your very, very beautiful simulations, but they were very cell-specific, or like within into cell-specific. But tumors, they are heterogeneous. And I wonder, how do you consider cell-to-cell communication and its effect on the simulations you're conducting?
Yeah, the short answer is we don't. So at the moment, we don't really consider cell-to-cell communications. We don't even really consider the heterogeneity of a tumor. I mean, a tumor is, right, it's incredibly complex. It has all sorts of different cells. Cells on the left side look different from the right side. So that's not really included yet. That's something we've thought about, and we are hoping to have, as I was mentioning, this modeling where you can select different regions, for example, and you can say, in this region, my tumor is different. It has maybe more differentiated, or it has become this weird blob with 5 chromosome 5s or something, who knows? Yeah, it gets very complex. We're having enough trouble at the moment with just normal cells or relatively normal cells, but those are all great questions that we'll hopefully address at some point. And if you have a good model for intercell interactions, we would be happy to include that somehow.
Yeah, thanks for a very interesting talk. So my research is on spatially fractionated radiation, and I was wondering whether your modeling and your investigations could be used to see how the areas of high doses interact with areas of low dose, and whether we would be able to simulate any of the differences in those areas as well.
Yeah, I'm thinking. Not really, I would say. If you have a hypothesis of how or what is happening, then we can always include that. We're modelers, we can include anything. But there's not, so the tissue at the moment is quite static. So all those effects, if you think it's basically just that you have, for example, if you think about grid therapy or so, then you could have some different, you can have a differential outcome in those different areas, but it's not something that is included like that, I would say. It's an interesting question, but.
All right, I think we are through our questions here. So Jan, thank you again very much for super cool stuff. And now we have a sound. Look.
Thanks for having me into all the great questions. Very tricky questions, but good questions.
Thank you, Jan. Thank you.
Hopefully next question.
All right.
Bye.
Thank you for joining.
Bye, everyone.
Bye now.